Next speaker is Rena Orman.
Hello everyone, my name is Rena Orman, as you just heard, and my research interest is in neurobiology of aging. I have special interest in several brain regions that I'm about to introduce you to, but before I do that, I want to introduce you to the character with top billing. Ladies and gentlemen, please meet the star of my 8-minute show, Carolia prespicillata, Shiba's short-tailed fruit bat. These fruit bats are really cute. They have gregarious personalities. They possess a number of remarkable features. We have a colony of these bats in-house. These bats are the descendants of wild-caught bats from the caves of Trinidad, almost like 30 years ago. Contrary to what is believed that bats are mice with wings, they are not. Bats are very closely related, these particular bats, are very closely related to whales, cows, horses, shrews, hedgehogs, carnivores, and scale anteaters. They weigh on average between 15 to 19 grams. Carolia typically has 2 reproductive periods per year, and the gestation takes about 120 days. It's 1 pup per liter, and the newborn weighs about a quarter of mother's weight. Mother carries it for about 2 months postnatally, and then it's flying. The age of sexual maturity for females is about 1 year, for males it's closer to 2 years. They are known to live up to 13 years in captivity. When living conditions are not optimal, shortage of food or the temperature is not to their liking, they can experience torpor, which is a short hibernation. If unpleasant living conditions persist, they can die. They roost in caves, hollow trees, buildings, typically roost in hair. Carolia exclusively, this particular Carolia, exclusively eats fruits, about 50 different species, and it has a very strong sense of smell and good vision. A number of features make Carolia an attractive model for biomedical research in general. They have exquisite control of autonomic functions. They have ability to bring their heartbeat from nearly 1,000 beats per minute to near zero in a very short period of time, which allows them to be a good model for studies on extreme survival conditions. Their reproductive biology, including the real menstrual cycle and placenta during pregnancy, is much more similar to that of humans than rodents are to humans. But most importantly for us, bats' brain architecture is a lot closer to humans than rodents are. And its longevity is priceless for a number of research aspects, including neurobiology of aging and neurodegenerative diseases. So neurodegeneration occurs in normal aging and is accelerated in disorders such as Alzheimer's and Parkinson's disease. An established convergence point for the different mechanisms of neurodegeneration and cell death is calcium regulation. Normal cellular calcium regulation is achieved through multiple mechanisms, including intercellular buffers and sensors, such as calcium-binding proteins. Losses of calcium-binding proteins can cause calcium dysregulation and lead to neurodegeneration. Calcium-binding proteins are a hallmark of inhibitory neurons throughout the brain. Regional losses of parvalbumin, calbindin, and calretinin in humans and animal models of Alzheimer's disease suggest that neuronal inhibitory mechanisms are impaired in aging and neurodegeneration. So you are looking at a, where is the, so you're looking at a set of sagittal sections that are stained for latexin protein in green, and double stained. Each section is stained with one of the calcium binding proteins. So, toleratement, albumin, and albumin. So the image, what this image reveals to us is A, that the claustrum is really large in the back, remarkably large. The specificity of latexin antibodies for claustrum and dorsal endothelium, can you see? Yes. Is very impressive. The pattern of latexin staining in claustrum allowed us to delineate claustrum subnuclei, subregions. There are only two other structures in the bat's brain that are stained so distinctly, and we will come back to it in a moment. The boundaries of claustrum and dorsal endothelium nucleus are also framed with by distinctive pattern of calcium-binding proteins. The main point to take away in this image, other than the fact that the image is gorgeous, is a reverse pattern for of staining for toleratement and probalbumin. And please follow. Can you follow it? Do you see the dark? Okay. So this is toleratement versus probalbumin. The calbindin is ubiquitously present everywhere. These images are a summary of our finding that there are at least 3 inhibitory systems that separately govern multiple claustral regions. All of these features can be easily accessed due to the claustrum size, large size, either anatomically or physiological studies in the back, either acute slices or in vivo. Okay. So here, I just wanted for a few seconds to illustrate the basic anatomy of dorsal hippocampal formation and unique advantages of the bat's brain using the new N and probalbumin staining as biomarkers. First, I want to call your attention to the CA3 region right here. Oh, okay. To the CA3 region, to the compact cell layer in area CA3, in contrast to the broader cell layer of the CA1. Very similar to human brain and completely opposite to rodents. Second, the bat brain allows us to see very clearly prosubiculum. The prosubiculum is a prominent feature of primate brains and is not visible in rodents. And the last literally 30 seconds, or I have a little bit, just 30. Okay. So for the last 30 seconds of my presentation, I will bring back the latexin staining in the brain and point you to the other 2 areas that stain very distinctly, which is paraventricular nucleus of hypothalamus, it's right here, and as supraoptic nucleus. So these 2 nuclei, well known, established structures as autonomic and neuroendocrine regions. Both are known sites of vasopressin and oxytocin releases. And we confirmed that both vasopressin and oxytocin are strongly expressed in Harolia, in PVN and SON. What we found, which is remarkable, is that vasopressin expressing cells are not uniform population. Let's see here. Some of them are expressing latexin, some are latexin positive cells, and some are not. So are the latexin positive cells excitatory neurons, as they are in claustrum, or is latexin expressed in neuroendocrine neurons as well? Lots of questions to answer. With these 3 short vignettes, I hope I convinced you that Karolia makes a very attractive model for the studies of neurobiology of aging and neurodegeneration, and that you will actually look at the bats now with different bats. Thank you.